Hi, my name is Peter Paré, and I'm happy to be giving the inaugural Here's to Your Good Health Pender Island Healthcare Society lecture here as part of the uh, library series. So I'll start by sharing my screen and my slides. So the title of this talk is Here's to Your Good Health, Your Genome Roadmap for Present and Future Health, question mark. Question mark is important because I'm not advocating that necessarily it is the roadmap to your present and future health, but that's what we'll discuss over the next little while. So what is the genome? Well, the genome is the entire set of DNA instructions found within the cell. And virtually all cells in your body contain this genome. So every cell has complete instructions, a whole set of instructions, the entire genome. It's not quite true exactly because the red blood cells circulating in your blood don't have any nucleus and therefore no DNA in them. But they did have DNA. They had the genome in their nucleus when they were in the bone marrow, and they just simply extruded the nucleus and the DNA before they exited the bone marrow and entered your bloodstream, because it's easier for them to function without the bulky nucleus within them. So virtually every cell has the entire genome within it. And that genome is contained in 23 pairs of chromosomes that are present within the nucleus and a small chromosome in the cell's mitochondria. So the genome contains all the information needed for an individual to develop and to function. So the next slide is a pictorial depiction of that. Here's a cell with the nucleus and mitochondria. And here are the 23 pairs of chromosomes that contain the genome. There's chromosome number 1 all the way up to chromosome number 2, and then there's the X and Y chromosome. So you get one of these pairs from your mother and one of these pairs from your father. And if you're a male, you get one X from your mother and the Y from your father. If you're a female, you get an X from both your mother and your father. So most of the genome is contained within the chromosomes, but there's a small amount of DNA and chromosome DNA sequence in the mitochondrial DNA. Now, interestingly, you get the mitochondrial DNA only from your mother because sperm contains no mitochondria, whereas the ovum that you get from your mother contains mitochondria. So you can use the mitochondrial DNA sequence to trace back your maternal line, whereas if you're a male, you can use the Y chromosome sequence to trace back your paternal line. So the human genome is pretty big. It consists of 3 billion letters, the A, T, C, and G. So if we type those out, if we had one page of A, T, C, G, etc., 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 then to type out the whole 3 billion letters would take a pile of papers 300 feet tall. So it's big, and it is composed of a sequence of only 4 letters, A, T, C, and G, which stand for adenine, thymine, cytosine, and guanine. These are bases as opposed to acids, and they're actually paired with their opposite numbers, as I'll show you. So A is always paired with T, T with A, C with G, and G with C. So there's actually 3 billion of these bases in a row paired with an additional 3 billion. So it's 3 billion base pairs long formed in a double helix, but of course divided into chromosomes. Now, gene is only a portion of the genome. So a gene is a small portion of the genome which has the sequence that spells out the instructions for making a specific protein. Proteins are the main structural and functional units of the body, and muscles are made of protein. Hemoglobin in your red blood cells is a protein. All the enzymes in your body are proteins, and all the antibodies in your body are proteins. So proteins are really the end product of the instructions contained in your genome. Humans have about 23,000 genes, which sounds like a lot, but actually those 23,000 genes make even more than 23,000 proteins, because some of the genes can be spliced and to make shorter proteins. So there's more than 40 or 50,000 different proteins that can be made from those 23,000 genes. But remember, size is not everything. And for instance, the humble COVID-19 virus contains only 30,000 letters in their entire genome and only codes for 12 proteins. So there's only 12 genes in the COVID virus genome and coding for 12 proteins, which make up this small but impactful organism. So size is not everything. So the letters A, T, C, and G can spell 64 different words. Go home and write it all out. A, T, G, G, A, T, etc. So 64 different words, and each of these 3-letter words codes for one of the 23 amino acids, which are the building blocks of proteins. So what happens is that a particular 3-letter word in your DNA sequence, for instance, AAA, is translated in the protein to the amino acid lysine, or AGA is translated into arginine. So long chains of DNA are coding long chains of amino acids, and linked up, those amino acids form proteins. So this is a picture of what a protein looks like, this long chain of amino acids, often folded up in very intricate designs. So the sequence of the human genome was first established about 2 decades ago. So in 1990, US Congress established funding for doing the first genome and set a target date for completion of 2005. So they anticipated that it would take about 15 years to complete one genome, just to get the sequence of one individual. In fact, they announced the completion in 2003, so it only took them 13 years to complete one genome. The cost, $2.7 billion in 1991 dollars. So expensive to get the sequence of one individual. Who was that individual? Well, we actually don't know. It's an anonymous individual. It may have been more than one individual. In any case, that sequence has become the reference sequence. And now you can get your whole genome done for the cost of $399. So 3.7 billion, now down to 399. Here's a graph showing what's happened to the cost per human genome. So this is going from 2001 up to 2021, so over a 20-year period. As I said, the original one cost $2.3 billion. By 2021 or 2001, one genome only cost $100 million. And then over the decade, 2 decades, it has dramatically decreased. This line would be an exponential decrease in cost because, as you can see, this is a log scale, 100 million, 10 million, 1 million, etc. So if the cost had followed this decline, it would be an exponential decline. In fact, the decline has even been more dramatic with this dramatic decrease in cost of doing genome sequencing in around 2007. And now, as I said, in 2023, the cost to do a whole human genome is around $399. So that's a dramatic decrease. The cost now is 1/22 millionth of the original cost. Fairly dramatic. So there are variations in the human genome. We're not all the same, and that's why it's necessary to do your own genome. So we're almost all the same. About 99.8% of the sequence in our genome is identical in all individuals, but we're all different. So at least 5 million letters in your genome are different than the reference genome. So take any 2 people in this room, as long as they're not related, there'll be at least 5 million spots in their genome where they differ from each other. And the most common difference is just the substitution of one letter for the other. So where most people have a C in the sequence, they could have an A. That's a mutation. Now, interestingly, there's not much difference based on ethnicity. This is a picture of my granddaughter, Ella, and her genome is about 0.2% different from this Asian child, whose genome is about 0.2% different from this African-American child. And similarly, there's about 0.2% difference between Ella and the African-American child. So not much variation. There's variation between ethnicities, but not a lot of difference between them. And similarly, there's not even much difference between different primate species. So here's Ella again. She's only about 1.6% different in her genome sequence from this baby chimp, and about 1.6% difference from this baby gorilla. And interestingly, there's as much difference in genome sequence between the chimp and the gorilla as there is between Ella and either of them. In fact, there's only about 8% difference in the genome sequence between the mouse and Ella, a little more between Ella and this fruit fly, about 56%. So the variations in the human genome are called mutations, and mutations have developed over many millions of years in the genome due to cosmic radiation and due to errors in replication of DNA in the chromosomes. So the most common, as I said, is a single substitution, whereas most people might have a C at a particular location. A minority of individuals have an A. So that's a single base pair mutation. There are also more complicated mutations where a whole short series of bases is missing. So here, where most people have a CTT at a particular location, a minority of individuals are missing that codon. That's what we call a 3 base pair sequence that codes for an amino acid. So this individual is missing this CTT at a particular location. That's called a deletion. In fact, this deletion is called delta 508 mutation, and it is the mutation that underlies cystic fibrosis. So 508 is the 508th amino acid in the cystic fibrosis protein. And individuals who have this mutation are missing one of the amino acids in that protein, and simply missing one amino acid causes the protein to get all misfolded up, and it doesn't function as it should. The normal protein is a chloride channel in cells and pumps chloride out of cells. And with this mutation, the protein simply doesn't function at all, and it results in cystic fibrosis. Now, since cystic fibrosis is a recessive disorder, you have to have two copies of the abnormal gene. So if you inherit a missing codon from your mother and your father, then you're likely to get, well, you will inevitably get cystic fibrosis. So there are carriers, people who are missing that codon from one of their chromosomes, but the normal chromosome can compensate completely, and so carriers have no problem. That's called a recessive disorder. A dominant disorder is where having the mutation only on one chromosome is enough to cause the disease. So Huntington's disease is an example of such a mutation. You can have one normal chromosome, but if you have one mutated chromosome with the Huntington mutation, you can get the disease. So variability in the human genome is responsible for lots of things. It's responsible for differences between individuals in traits such as hair color, height, eye color, all the things that make us look different, act different. So variability is responsible for differences between individuals, differences in our response to environmental factors, and diseases. So diseases or disease susceptibility. So they can cause actual diseases or increase your risk of specific diseases. In fact, diseases caused by genetics can be divided into two major categories. One are called single gene diseases. In a single gene disease, a single mutated gene, a substitution of a letter or a missing codon, is sufficient to cause the disease because it has a big effect on the protein that that gene codes for. These are rare disorders, and they're rare probably because they have a significant effect on the organism, and therefore that organism is less likely to procreate. So evolution has tended to make these kinds of mutation, and therefore these diseases relatively rare. They're rare, but there are many examples of them. So here are some of the examples of the approximately 10,000 single gene disorders that have been identified. So cystic fibrosis, I've already mentioned, is rare. It only occurs in 1 in about 3,000 births among Caucasians. It's rare, even much, much rarer in Asians or Africans. Sickle cell disease is more common, also a recessive disorder occurring in about 1 in 400 African Americans or Africans from West Africa anyway. Huntington's disease, a dominant disorder I mentioned, is much rarer, occurring in only 1 in every 35,000 births. And then hemochromatosis is fairly common, but a relatively mild disease, occurs in 1 in about 300 births. So all told, 1 in about 100 births have some kind of single gene disorder. So they're relatively rare. So the other kind of diseases caused by mutations in genes are complex genetic diseases. So the difference here is that multiple mutated genes are necessary to give you susceptibility to these diseases. And that's because there's only a small effect of these mutations on the protein that the gene codes for. And because environmental factors are also necessary to cause the disease. So simply having these mutations doesn't inevitably give you disease. It has to interact with some environmental factor, something you eat, something you smoke, something else in your environment that interacts with the genetic susceptibility to give you the disease. So they increase susceptibility rather than definitely causing the disease. And they're common. I mean, these are the common diseases that affect all of us. And here's a list of them. Atherosclerosis, which causes heart attacks and strokes, type 1 and 2 diabetes, Alzheimer's disease, schizophrenia, Parkinson's disease, inflammatory bowel disease, cancer, asthma, COPD. COPD and asthma are the two that I worked in my career. All of these have a genetic component. They run in families. They have a contribution of genetics to them, but environmental factors, lifestyle factors influence whether those susceptibility genes actually result in disease. So you can't think of them as the cause of disease. They're simply susceptibility factors. Now, what else can you learn by looking at your genome besides disease susceptibility? Well, you can learn things about your ancestry. You can determine whether you have a risk of a rare genetic disease in you or whether you're a carrier and then have the possibility of passing on that susceptibility or that disease to your offspring. You can learn about susceptibility to these common diseases. And you can also learn something, and increasingly more, about how you will respond to specific medications. So this is a field called pharmacogenetics. And some of your genome can predict how efficient or efficacious certain drugs are for you. As you know, some people respond well to certain drugs. Some people don't respond well at all. And a lot of that is dependent on your genome. Similarly, side effects from specific mutations are genetically determined. So some people develop severe or mild side effects from some medications. Others develop no side effects, and that is largely related to your genetics. So why not have a look at your genome? Well, a few years ago, I did have a peek at mine, so I'd like to tell you a little bit about that. Now, I didn't get my whole genome sequence because it wasn't available at the time, or it was costing in the thousands or tens of thousands of dollars. So companies such as 23andMe and Ancestry.ca came up with partial genome sequences. So you could send them your DNA and you could get it sequenced maybe 1 or 2 million of the letters in your genome. Now, that doesn't sound like a much, but they selected those 1 to 2 million spots in your genome to be enriched for variable sites between individuals, sites that contribute to different traits and contribute to different diseases and disease susceptibility. So although it's much less than 1% of the genome, it contained what you could get sequenced from 23andMe and these other companies was more than 1% of the information in the genome. And I couldn't resist when I saw an ad online for 23andMe that showed me that I could find out about how DNA could help me plan for important things in my life, how it could upgrade my health records with my carrier status, help me live well at any age. You can see how happy this individual is, and get treatment that's right with you. That's the pharmacogenetics aspect. What really sold it to me, of course, was when they had a holiday special. So It was $499 to get this done. But then just before Christmas I think it was 2016, they offered a discount at $99. I couldn't resist. So I sent off, and they sent me a little package with a mouthwash, did my mouthwash, sent it back. And about 2 weeks later, I got my results online. And I continuously, ever since then, been getting updates to what my genome predicts. So here are some of the things that 23andMe will tell you about. So you go to your own little website, and it'll give you something about your health and something about your ancestry. So let's start looking at my ancestry. So my paternal line, and this is what they've traced back, is where your DNA originated about 500 years ago, because there's been so much mobility of humans since that time. This tells you where your DNA originated about a half a century ago, or half a millennium ago. So it wasn't any surprise to me that my paternal DNA came from Northern Europe here, my French name, so probably from France. In fact, I knew that my great-great-great-great-great-great-great-grandfather came from Normandy. So I wasn't too surprised by that. But my maternal DNA from the mitochondria was more interesting. As you can see, blue means more likely it's where it's from. And as you can see, my maternal DNA largely came from, or is likely to have come from, northern Scandinavia. So up here in Norway or Sweden. That was a bit of a surprise to me. I thought my grandmother and my maternal line came from Ireland and Scotland. But the explanation must be that my great-great-great-great-grandmother was a Viking, which may explain some of my behavior. So, well, the other thing they can tell you about is how much Neanderthal you are. So my DNA is about 3% from Neanderthals. So Caucasians, not Africans, but Caucasians have a substantial part, well, 3% of their DNA can be from Neanderthals. And I'm at the 88th percentile. So the average user of 23andMe has about 2.7% of Neanderthal DNA. So I'm over the average for that. You can also find out some DNA relatives. So they report to you other people who have had their DNA sequenced by 23andMe. Now, you actually have to allow them to broadcast this, but I did that, and I've now found more than 1,500 relatives, mostly distant relatives, but I found that a couple of my nephews had their DNA done by 23andMe, and I have multiple first cousins once removed. So these are daughters or sons of my cousins who have had their DNA sequenced. In fact, amazingly, I found a first cousin once removed that I didn't know existed. She was adopted, and she sent her DNA to 23andMe, presumably because she was curious about her origin. And it turns out she is related to me. So I actually contacted her. So you can put permission to be contacted into this site. And I said by email, I asked her whether she wanted to delve into her origins more, because I could look into it. And she decided not to. It's very interesting. She must have been interested in her origin to have sent in the DNA in the first place. But then when it came to it, she said she had had a happy life. She was happy with her family. She didn't want to find anything more. So besides ancestry, you can find out about some of your medical propensities. So there's clinical reports. So some of them are trivial. These list a list of traits that you likely have based on your DNA. So some of them like the alcohol flush reaction, whether you have bitter taste perception, your earwax type, your eye color, or whether you have curly hair. Well, I found that I'm not likely to have the flush reaction when I drink alcohol, which I knew, and then other useful information like my earwax type is wet rather than dry. My eyes are likely blue. In fact, they're bluey green. And surprise, surprise, I likely to have straighter hair than average. That is, if I had any hair left. So among the clinical reports are your risk of having type 1 diabetes, atrial fibrillation, venous thromboembolism, type 2 diabetes, age-related macular degeneration, and a whole list of other complex genetic diseases. So let's just look at one of these. What it shows you is a bar graph of your chances of getting these diseases relative to the average. So for age-related macular degeneration, I have a lifetime risk of 5.3% of getting macular degeneration, whereas the average person has about a 7% chance, so a slight decrease. Interestingly, there are also some reports that before you click, it says, this is a locked report. So before you click on it, you get some education of what it means. So if I clicked on this one, some text comes up and says, genetics can substantially affect your Parkinson's disease risk. You're about to learn, if you click on the next button, whether you have a relatively rare mutation in the LRRK2 gene that raises your lifetime Parkinson risk to more than 50%, compared to what the population average is between 1 and 2%. So as I said, Parkinson's disease is a complex genetic disease for the most part, but there are some rare mutations that increase the risk similar to a single gene disorder, so a 50% risk of developing the disease. So that what they're saying here is that if you want to find out if you have this risk, press on the button. If you don't press on the button. So actually, I didn't press on that button, so I don't know if I'm a carrier of this rare disease, Parkinson's disease gene, because what's the point? There's no present treatment to prevent Parkinson's disease. So to know or not to know. Okay, so other important things you can learn from your genome is some of your drug responses. So here are some drugs. What I found that I have increased sensitivity to warfarin, or also known as Coumadin, which is a blood thinner. So if I ever needed a blood thinner, something to reduce my chances of a stroke or a heart attack, then I could tell my physician that I have increased sensitivity and they might reduce my dose. And here, for these other drugs, I had a typical sensitivity and efficacy. So there was nothing there to worry about. Now, you can still get your genome partially sequenced by 23andMe. And in fact, just 2 weeks ago, they had a Valentine's Day offer with 40% off for getting your DNA done by 23andMe. So here you go, 40% off. Show your health some love. Get health and ancestry and $209 instead of $249. But more exciting is the fact that you can get your whole genome sequence done. So this is, as I said, only a partial sequence. There's now companies including sequencing, outsmart your genes. And there's the ultimate DNA test. So whole genome sequencing. You can get the $399 version, or if you want it really fast, expedited, you can get the $1,999 sequence. They're the same. This one just comes faster. So you can get results within 8 to 12 weeks, and they offer privacy forever. They won't sell or share your DNA with anyone. I'm tempted, but I haven't sent in my DNA yet. So, should you do your DNA sequence? Should you do your genome sequence? In fact, a question arises, should we do genome sequencing in everybody? These are important questions, and I don't have answers. But there are some downsides to doing your genome sequencing. The risks imparted by genome variants are really low. So they're, for most part, right, there's the single gene disorders, which are pretty rare. And by your age, you probably, most of us know whether we have them or not. So there's not much use of knowing about those single gene disorders unless it is to tell your kids or grandkids about them. And for the complex genetic diseases, the risks are low. And actually knowing your genome sequence can cause some false sense of security or some excessive worry. So if you have some gene variants that increase your risk of something, you can get worried, or if you don't have them, you might get a false sense of security. Also, there's the potential that having your genome sequence out there could compromise your privacy or insurability. So if you're planning to leave any of your body fluids at a crime scene, I wouldn't get your DNA sequenced. And often what you can learn doesn't help you, in the sense that there's nothing that can be done to alter the risk, like, for instance, Parkinson's disease, which I mentioned. In addition, there's no capacity in our healthcare system or within our society for the amount of genetic counseling that would be required if everybody went out and did their DNA sequences. It's not easy to interpret all these results, and people would be flocking to their doctors to ask what this means. Now, I used to list cost as a downside to doing your genome sequence, but as you saw, that is no longer pertinent. It's only $399. So I think we have to start living with it. So like it or not, I think whole genome sequencing is coming. So some countries, including UK, France, and Australia, are aiming to provide free DNA sequencing through their healthcare systems. It hasn't started yet, but it's in the planning process. In the UK, they're presently doing a study where they're doing whole genome sequencing on 100,000 newborns. So they're getting DNA as soon as the infants are born, and they're doing whole genome sequencing. But interestingly, in this study, they're only going to look at the 200 single gene disorders that are potentially treatable. As I said, there's about 10,000 now known single gene disorders. Most of them are not treatable, but there are 200 where something can be done, some medicine or some avoidance of environmental factors can help alleviate the disease. So they're only going to look at those sequences for those 200 diseases, and until something is known about the others, not look at those. So whole genome sequencing will likely become a routine part of medical assessment in the not too distant future. And there's a whole science of this called personalized medicine. So I think, like it or not, like the internet, like social media, like artificial intelligence, I think we have to learn how to deal with whole genome sequencing. So to end, learn to love your genome. You're my base pair. So I'll sign off now. In the library, I'll be answering any questions that are posed. But you can always email me if you have questions. And my email address is peter.paré, P-A-R-E, at hli.ubc.ca. So I hope you enjoyed this talk, and send me any questions you have. Thanks a lot. Bye now.